ever feel like you're just drowning in data, but the really key insights feel buried, like miles deep.
Happens all the time. Mountains of information, yeah.
Well, today we're doing a deep dive into a book that tries to help with exactly that. Elegant SciPy.
Right. It's all about writing, well, effective scientific code using Python, SciPy, and NumPy libraries.
And the focus is really on clarity, being concise, and efficiency, getting code that just feels right, you know?
Exactly. And for you listening, think of this as a shortcut.
Yeah.
We're pulling out the core concepts from Elegant SciPy, the stuff you really need to understand how these tools can, like, boost your scientific work.
We'll cut through the noise, give you the essentials with some solid real-world examples.
That's the mission.
And what I liked about the book, it's not just dry code examples. It argues that writing good scientific code is actually, well, creative.
Like crafting a story, almost.
Yeah. It covers using NumPy and SciPy for all sorts of things: gene expression, images, signals, optimization.
We're really wide range.
And it keeps coming back to this idea of elegance. It even quotes this piece from the New York Times comparing elegant software to, like, Virginia Woolf's writing.
Which sounds a bit out there, maybe, but the point is that it's not just about speed, it's about understanding the problem deeply. When the code feels right, it often means you found a better, more fundamental solution. It's satisfying.
Okay, I get that. So today, we'll extract some key principles, look at some cool examples from the book.
Yep. Give you a taste of what working elegantly with SciPy and NumPy actually means.
All right, let's dive in. Where does the book say all this power starts? What's the foundation?
It all starts with NumPy, and specifically NumPy's n-dimensional array, the end array.
That end array, okay.
That's the absolute bedrock for pretty much any numerical work you'll do in Python with these libraries.
Got it. And the book uses gene expression analysis as an early example for this end array, right? How does that work?
Yeah, think of gene data like a big spreadsheet, basically. Each row could be a gene. Each column, a different sample, maybe different tissues, different patients, whatever.
Right.
That structure maps perfectly onto a 2D AND array, a matrix. And each little cell in that matrix holds a number, like how many times you counted that gene's RNA in that specific sample.
Okay, so it's a neat grid for holding lots of numbers, but why underarrays? Why not just use standard Python lists? What's the big deal?
Speed. Simple as that.
Speed.
Massive speed increases. It comes from something called vectorized operation.
Vectorized, okay.
With NumPy, you can do math on the entire array at once. You don't write Python loops to go through element by element.
Okay.
Under the hood, these operations are often running in super optimized C code. So if you need to add 5 to a million numbers in an array, one NumPy operation, boom, lightning fast. Try that with a Python list, looping a million times, way, way slower.
Right. That makes total sense, especially with the huge data sets in science.
Exactly. It's critical.
The book shows multiplying a whole array by one number, just like that. Super efficient. Now, they also talk about broadcasting. What's that about?
Broadcasting. Yeah, it's another really powerful NumPy feature.
Okay.
It lets NumPy perform operations on arrays even if their shapes aren't identical, as long as they're compatible.
Compatible how?
Well, NumPy can sort of intelligently stretch or duplicate the smaller array across the dimensions of the larger one behind the scenes.
Okay, so you don't have to manually reshape things all the time?
Precisely. It avoids a lot of explicit looping or resizing code. The classic example is the outer product of two vectors.
Outer product?
Yeah, if you have a column vector and a row vector, broadcasting lets NumPy automatically create the full matrix where each element is the product of the corresponding row and column elements. Very neat, very efficient.
Got it. Speed from vectorization, flexibility from broadcasting. Okay, so moving on to gene expression, again, normalization. Why is that so important?
Well, the raw counts you get from sequencing experiments, they can be misleading.
How so?
Lots of technical things can affect them. Like maybe you just got more sequencing data overall for one sample compared to another. Higher counts across the board, but it doesn't mean the biology is different.
It's the library size difference, right?
Exactly, library size. And also, longer genes just naturally tend to get more reads than shorter genes, more sequence to, grab onto.
Okay, so raw counts aren't comparable directly. We need to adjust.
Exactly. You need to normalize to account for those biases if you want to make fair comparisons between genes or between samples.
And Elegant SciPy introduces RPKM for this. Reads per kilobase.
Per million mapped reads. Yeah. RPKM tackles both those issues we mentioned.
Wow.
The per million mapped reads part scales things based on the total reads for that sample that handles the library size difference.
Okay.
And the per kilobase of transcript part scales by the length of the gene itself.
Right, accounting for gene length.
So RPKM gives you a number that's, in theory, adjusted for both sequencing depth and gene length. Makes comparisons more meaningful.
Okay, RPKM for correcting known biases. But then the book mentions quantile normalization. What's different there?
Quantile normalization is a bit more aggressive, maybe.
How so?
Instead of just adjusting for known factors like library size, it forces the entire distribution of expression values to be the same across all your samples.
The whole distribution, wow.
Yeah, it basically ranks the genes in each sample by expression, then sets the expression value for each rank to be the average expression at that rank across all samples.
So every sample ends up with the same statistical shape.
Exactly. It's useful if you care more about relative changes within your population of samples rather than absolute levels. Makes a big assumption, obviously.
That the underlying distribution should be similar.
Right. But the book points out it can be really good at highlighting subtle, consistent changes across samples that might otherwise get lost.
Interesting trade-off. Okay, let's switch gears. Image processing. How do numbers and arrays relate to pictures?
It's actually pretty direct. Think about a black and white image. It's just a grid of pixels, right? Each pixel has a brightness value. That's just a 2D NumPy array.
Ah, okay.
Yeah.
Each number is a pixel intensity.
Exactly. And for a color image, say RGB, red, green, blue.
Yeah.
You just stack 3 of those 2D arrays, one for red intensity, one for green, one for blue. So a color image becomes a 3D NumPy array.
Height, width, and color channel. Makes sense.
Totally. The book even shows creating random noise with NumPy and displaying it as an image using Matplotlib. It's that direct.
And they load a real photo, too, those Roman coins, using scikit image.
Right. And once that image is a NumPy array...
You can slice and dice it?
Exactly. NumPy's array slicing is super powerful for images. The book has this great example, adding a bright green square onto an image of an astronaut.
How?
Just by selecting a specific range of rows and columns in the array, selecting the green color channel slice for that region, and assigning it a high value, like 255.
Just assign a number to a slice of the array, and boom, green square on the astronaut.
Yep. It really shows how intuitive manipulating images can be once they're under arrays.
Very visual. Okay, they also mention image filtering. Is this where SciPy comes in more?
Yeah, SciPy's image module is packed with image processing tools, and filtering is a big part of that.
What is filtering exactly in this context?
Think of it like modifying each pixel based on the values of its neighbors. You apply a small pattern called a kernel or filter.
A kernel?
Yeah, across the image. This kernel defines a little calculation, maybe an average, maybe something more complex using the pixels it's currently over. The result of that calculation becomes the new value for the center pixel.
Okay, so you slide this kernel over the whole image?
Exactly. It's used for all sorts of things, blurring an image, sharpening it, detecting edges. It's a core concept from signal processing, really, applied to 2D image signals. Like a Gaussian blur just averages neighbors with certain weights.
Got it. So NumPy for the basic array structure and manipulation, SciPy's and Image for more advanced stuff like filtering. Okay, next up. Graphs and networks, using network X.
Right. Graphs are just ways to represent relationships. You have nodes, the things.
People, computers, genes.
Whatever. And edges the connections or interactions between them.
Okay.
Network X is the go-to Python library for working with these graph structures, creating them, analyzing them.
And the book uses the C. elegans connectome, the worm brain map.
Yeah, a fantastic example. A map of every neuron and synapse in that little worm. That's a network, a graph.
So what can you learn by analyzing that worm brain graph?
Well, one thing they look at is betweenness centrality.
Betweenness centrality sounds important.
It is. It measures how often a specific node, a neuron, in this case, lies on the shortest path between other pairs of nodes in the network.
So nodes that are on lots of shortest paths are kind of crucial intersections.
Exactly, like major hubs for information flow. And the book points out that neurons in the worm known to be involved in sensing touch, like AVAR and AVAL, actually have high betweenness centrality scores in the connectome graph.
Wow. So the graph analysis reflects known biology.
It can, yeah. It helps identify potentially key players.
Cool. They also mentioned strongly connected components. What are those?
In a directed graph where connections have a direction, like signals flowing one way between neurons.
Okay.
A strongly connected component, or SEC, is a group of nodes where you can get from any node in that group to any other node in that same group, just by following the directed edges.
So like a tightly knit cluster where everything connects back eventually?
Pretty much. Analyzing the worm connectome showed its largest SEC was actually smaller than you'd expect if the connections were just random.
Meaning?
It suggests the network isn't just one big tangled mess. It might be more modular, maybe with specialized groups of neurons that are tightly connected internally, but less so globally.
More segregated processing units. Interesting. Now, they also link graphs back to images with region adjacency graphs, or RABs.
Yeah, that's used in image segmentation after you've divided an image into different regions.
Like finding all the distinct objects.
Right, or just areas with similar color or texture. The R edge represents those regions as nodes.
Okay.
And you draw an edge between two nodes if their corresponding regions in the image are touching, if they're adjacent.
So it captures the spatial layout of the segmented regions.
Exactly. Then you can use graph algorithms on the R-RAID to analyze how the image parts are connected, maybe merge small regions, stuff like that.
Clever. Okay, next up. The fast Fourier transform, FFT. This one always sounds intense.
Yeah, it has that reputation. But the core idea is actually super elegant.
Okay, break it down.
The FFT is just a really, really efficient way to take a signal. Could be sound waves changing over time or pixel values changing across space in an image.
Yeah.
And break it down into the frequencies that make it up.
Frequencies, like musical notes in a chord.
Exactly like that.
Yeah.
A complex sound is just a mix of different simple sine waves, different frequencies. The FFT tells you which frequencies are present in your signal and how strong each one is.
Okay, so it translates from the time or space view to a frequency view. Why is that so useful?
Because patterns that are totally hidden in the original signal can become really obvious in the frequency domain. The applications are everywhere. Audio processing, JPEG image compression, Wi-Fi signals, MRI scans, countless things rely on it.
And the fast part, is it just faster than other ways?
Dramatically faster. The straightforward way to calculate this, the discrete Fourier transform, or DFT, gets computationally very expensive as your data gets bigger, like n-squared operations, where n is your data size.
Okay.
The FFT finds clever shortcuts to get the exact same result using only n times log n operations.
N log n versus n squared. That's a huge difference for large n.
Astronomical.
Yeah.
It's what makes analyzing large signals practical on normal computers.
So how does the book show this off?
They have a great radar example. Imagine radar bouncing signals off things.
Okay.
The raw signal coming back over time can look like a complicated mess, especially if there are multiple targets.
Right. Lots of overlapping echoes.
But if you take the FFT of that messy time signal, suddenly peaks appear in the frequency domain corresponding to the ranges of the different targets.
Whoa, so the distance shows up as a frequency spike?
Sort of, yeah. The FFT transforms the time domain echo data into a frequency spectrum that reveals the distance information clearly. They mentioned using it to detect a faint echo from a mine wall that was basically invisible in the raw signal.
That's powerful, seeing the unseen signal. Okay, moving on. Contingency tables and information theory. Sounds statistical.
It is. A contingency table, you might know it as a confusion matrix.
Ah, right, from machine learning.
Exactly. It's just a simple table to compare two ways of categorizing the same things. It counts how often each combination of labels occurs.
Like predicted spam versus actual spam.
Perfect example. The table shows you true positives, correctly called spam, true negatives, correctly called not spam, and then the errors, false positives and false negatives.
So it summarizes classifier performance. Where does information theory fit in?
Information theory gives us ways to measure, well, information or uncertainty, randomness.
Like entropy.
Exactly. Entropy is key. Elegant SciPy introduces a metric called variation of information, SIGAR. Yeah, it uses entropy and related concepts like conditional entropy to measure how different two labelings or two segmentations of the same data are.
Like two different ways of segmenting an image.
Precisely. Psi gives you a single number based on information theory principles. A low V means the two segmentations are very similar. Knowing one tells you a lot about the other. It's a more principled way to compare them than just counting mismatched pixels.
Interesting. Using entropy to measure segmentation similarity. Okay, next. Function optimization with SciPy. Finding the best settings for something.
That's the core idea, yeah. Finding the minimum or maximum of some function. SciPy's optimize module has a bunch of algorithms for this.
And the book uses image registration, aligning images.
Right. Say you have two photos the same thing, maybe slightly shifted or rotated. You want to line them up perfectly.
How does optimization help?
You define a cost function that measures how badly aligned the images currently are.
Like how different the pixels are in overlapping areas.
Exactly. The book starts with mean squared error, MSE, between pixel values. Lower MSE means better alignment.
Okay.
Then you use a SciPy optimization algorithm. It tries adjusting the alignment parameters, the shift, the rotation iteratively, trying to find the parameters that minimize that MSE cost function.
So the optimizer searches for the best alignment based on minimizing the error.
Yep. And they show a more advanced cost function too. Normalized mutual information, NMI.
NMI, why use that?
It's apparently much better when the images might have different brightness or contrast, or especially for multi-channel images like aligning the separate red, green, blue channels of old color photos.
Like those Prokudin-Gorsky photos.
Exactly that example. They show NMI producing a much clearer, better aligned final color image from those separated plates compared to just using MSE.
So the choice of cost function is critical.
Hugely critical. And the book also mentions pitfalls, like getting stuck in a local minima, finding a pretty good alignment but not the absolute best one, and techniques like basin hopping to try and avoid that.
Right, finding the true global minimum. Okay, last big topic. Handling big data that won't fit in memory, using the tools library. Very relevant today.
Extremely relevant. Tools is all about functional programming techniques for processing data streams efficiently.
Streams. Like processing data as it flows through, not loading it all first.
Exactly. It uses lazy evaluation. Data is processed piece by piece, element by element, only when needed.
So you can handle files bigger than your RAM.
Way bigger. You never hold the whole thing in memory at once.
How does tools make this easy?
It provides functions for building up pipelines of operations on these streams. The book highlights the pipe function.
Pipe.
Yeah, it lets you chain operations together really clearly. You pipe the data stream into the first function, its output gets piped into the second, and so on. Very readable.
And the example they use is pretty wild.
The fruit fly genome, yeah.
Yeah.
Building a Markov model from the entire fly genome sequence.
On a laptop.
On a laptop, using tools. Processing gigabytes of DNA sequence, character by character, applying transformations, counting transitions, all streamed, using very little memory. It's a fantastic demo of elegance and efficiency for big data.
That's seriously impressive, just a few lines of tools code.
That's the claim. They also mentioned currying.
Currying? Like the food?
Yeah, no. It's a functional programming thing where you can take a function that needs multiple arguments and create a new version where some arguments are already filled in. Makes building flexible processing steps easier.
Okay, interesting technique. Wow, we've covered a lot of ground from Elegant SciPy. What's the main takeaway here?
I think the big message is about the mindset. It's not just about using SciPy and NumPy, but using them elegantly. Aiming for code that's clear, efficient, understandable, and often that leads to better science.
Right, the elegance isn't just for show, it reflects deeper understanding and often leads to more robust solutions.
Exactly, and the book gives you so many concrete examples of how to achieve that across so many different scientific areas.
Yeah, the range is incredible. Bioinformatics, image analysis, signal processing, networks, optimization, big data. It really shows how central these Python tools are.
They form the foundation for so much modern scientific computing.
And like you said, this deep dive is just scratching the surface, right?
Oh, yeah. The book goes deeper, has exercises, more advanced topics. If this stuff interests you, definitely check it out. Think about how these ideas apply to your work.
Definitely. So maybe the final thought is that striving for that elegance in code, as the book suggests, isn't just about being neat. It helps you think better about the problem itself.
Well said. You can unlock more creative approaches and ultimately lead to more impactful results.
So keep exploring resources like Elegant SciPy to really master the art and science of Python for scientific work.